morning, everybody. Just to introduce myself to people that just joined us, my name is Hassan, and I'm a postdoc at the University with Nicole Gerardo. But for today's talk, I'll be talking about the final chapter of my PhD, which was with Martin Kaltenbrot at the Max Planck Institute in Vienna, Germany. Specifically, I looked into the mutualistic association between cassida beetles, so wheat beetles, and their bacterial endosymbionts. But before I start and examining the questions that we pursued, I'd like to take a step back and sort of stand on the shoulders of two giants that use cassida as a system to explore questions in ecology and evolution. First, of course, is of the late Thomas Eisner, who in the 60s published a really cool study looking at the role of the fecal shield that the larvae carry obliquely throughout their development, which is basically to fend off predatory attacks by ladybugs and ants. But perhaps most relevant to today's talk is an even older study, one that just turned 80 years old, which is from a German zoologist by the name of Hans-Jürgen Stammer. And what Stammer did was basically utilize a ton of beautiful histology experiments to describe what he thinks was the first association between leaf beetles and symbiotic bacteria. Perhaps the biggest take-home message from this 80-year-old paper is that he describes these structures that are the anterior pole of the eggs that get oxidized, are involved in the vertical transmission of symbiotic bacteria. And so this was sort of our starting point and our launch pad for the system. And we started first by obviously characterizing this so-called symbiotic organism that was within this beetle. We weren't sure if it was a fungi or a bacterium, but when we looked at the bacterial community, we found that there's across 4 populations of Cassida rubiginosa, whether it's from the US, Japan, Germany, and New Zealand. They're dominated by one primary endosymbiont, one that clusters within the gamma proteobacteria, most specifically within the so-called symbiotic clades, alongside other primary endosymbionts of aphids, beetles, ants, as well as stink bugs. And of course, we wanted to know if we wanted to localize the symbionts across host anatomy. And what we were able to find is we utilized fish. I guess we're going to start here and ask if everybody can hear me. Everybody can hear me, okay. So we utilized fluorescence and pseudo hybridization to localize our microbe, using first Dalby counterstain as a non-specific fluorescence. And what you're looking at here is a whole body anatomy with dissection occurring right below the mouthpiece and going all the way down to the ovipositor. And we did this dissection using a female that was just in the middle of laying eggs, just so that we could see the transmission cycle, or visualize the transmission cycle of the symbionts as well. And so when we contrast the Dalby counterstain with a green probe targeting the primary endosymbiont, we find that symbiont localization is restricted to 2 parts. First, in the foregut, a pair of symbiotic organs on either side of the foregut, as well as this really crazy fluorescence that occurs in these accessory glands associated with the ovaries. And so when we take a closer look at the foregut, we find that the foregut is very similar to so-called bacteriomes that you find in aphids and tsetse. But unlike these bacteriomes, the symbionts are extracellularly localized. So even though there's a complete encapsulation, complete compartmentalization of the symbiotic community, the symbionts are nonetheless extracellular, which is unique and kind of surprising for us. And this is also the case when we took a closer look at these accessory glands that are associated with the ovaries. Again, there's a really dense symbiotic population associated with these finger-like structures, but the unifying feature is that the symbionts are nonetheless extracellularly localized. And one thing I wanted to note from this figure here is that these fingers, or these accessory glands, are connected to the ovipositor through very thin tubules, and that kind of funnel the symbionts towards the ovipositor while the eggs are being pushed out. And what you can see here is that the symbionts get further packaged into smaller vacuoles, as you can see right here. And what I ask you is to take a kind of a mental note of these small vacuoles, because they'll be very relevant for transmission. Symbiotic transmission. So we kind of went back and assessed Stammer's hypothesis that these so-called egg caplets are responsible for the vertical transmission of symbionts. And we took a closer look at the caplets. We utilized a couple of techniques, 3D reconstruction as well as fish, to look at what's in there. And when we reconstructed using the program in Mira over 1,000 slides, we were able to find that within each caplet are roughly 20 to 30 small vacuoles. And when we subject these small slides, or these cross-sections to fluorescent pseudo-hybridization fish, we found that these vacuoles are absolutely stacked or packed with the symbionts. And so this was really exciting for us because this provided us with an extracellular route of transfer of the symbionts. And so potentially we wouldn't have to rely on antibiotics to manipulate the association. Basically, we wanted to compromise the egg caplet integrity to disrupt the transmission route of the symbionts. And so, in doing so, we used a really low-tech technique to do so, which is basically poke the egg caplet. And easily enough, it worked. So I just poked a lot of egg caplets. And after poking the egg caplets, we would basically put a really small droplet of ethanol, and that completed the sterilization procedure. Yep. So having that experimental framework in mind, we started with 3 experimental groups to test symbiont function. First, of course, is an untreated control compared to an egg caplet that's been pierced as well as sterilized. But also we had a third group, which was basically an egg caplet that was not pierced, but nonetheless sterilized. So that you make sure that whatever effect that we find in this treatment here is not due to the fact that you inundate an egg in ethanol. And ideally, you want these 2 groups to have the same developments and fitness. And so when we look at the hatching rate across the 3 groups, we find that there's no statistical difference. Nonetheless, when we look at development and survivorship, we find a huge difference in the treatments where the egg caplet was pierced and sterilized relative to the 2 control groups, suggesting that the symbionts are in fact mutualistic in nature. Of course, the question is, what are they doing? And so here we combined genomic as well as transcriptomic techniques to assess symbiont function. And when we looked at the genome, this was perhaps the most surprising part of the study. The genome was tiny for an extracellular symbiont. It was about 4.25 MBs with a really, really low GC content at 15.4%. And just to give you a framework of comparison, the smallest known extracellular microbe is Mycoplasma genitalium, which is roughly twice the size of this genome for Cassid reviginosa, or the symbiont of Cassid reviginosa. So it's a really tiny genome for a symbiont that does not occur intracellularly within a bacteriocyte like Mira or Carcinella rugiae. And so this was surprising to us because still they have to contend with a fair amount of metabolic requirements that are not available in an extracellular matrix. And when we look at structural features of our genome, we do a 3D plot of GC content, genome size, and number of genes. We find that our symbiont falls strictly within the intracellular symbiont category, especially among ones that have undergone drastic genome reduction. Of course, given the phylogenetic position of our microbe, we rushed to annotate genes involved in essential amino acid biosynthesis and B vitamin biosynthesis, but there was nothing. And so this was kind of a shock and a depressing day in the lab. But it was good because it kind of, we had to take a step back and reassess the feeding ecology of our beetle and the nutritional requirements that these leaf beetles have, as well as the localization of the microbes. It's a really weird place to have a bacterium. It's at the foregut. Most nutritional endosymbionts that provide amino acids or B vitamins are in the hindgut or in organs that are completely separated from the gut. These microbes are in the foregut, kind of at the forefront of where digestion happens. So we hypothesized that the microbes might be involved in the degradation of complex plant polymers. That's exactly what we found. These are obligate folivores, the leaf beetles that we're dealing with. In order for them to access the nutrient-rich cytosol of plant cells, they must break through the formidable barrier of a plant cell wall, which of course is composed of pectin, hemicellulose, and cellulose microfibrils. But we found enriched in this tiny, tiny genome were genes involved in pectinase, or pectin degradation, which is, we were able to annotate a gene for pectin degradation, specifically pectinase of the polygalacturinate variety, which hydrolyzes pectin into galacturinic acid. monomers. And this was, and when we looked at the close relatives of our symbiont, we didn't find the enrichment or presence of this gene in any of the so-called symbiotic weight. And so we then wanted to ask globally, so the gene is enriched in the genome, but what is it in fact expressed? And so we looked at the global transcriptome of the sleep beetle, specifically the symbiotic organ, as well as the whole body. And we didn't just target this one pectinase that we found in our genome, we looked at all pectinase expression patterns. Nonetheless, we still find a huge upregulation of pectinase in the symbiotic organ, and all of it matches to our symbiont. There's a small expression of pectinase within the whole body, but they could be annotated, or we could map them to other gut bacteria that are present within the beetle. And so the gene is there, it's being expressed, but is it functionally conserved? And in doing so, we wanted to test the activity levels of pectinase, and we did a very simple protein extraction from the symbiotic organ. And then we inserted that protein extraction into a pectin-enriched agar plate and looked at the clearance zones. What we can see here is that the symbiotic organ has a really, really strong pectinase activity, suggesting that the gene is not just expressed, but it's actually also functionally conserved and active. And so with that, I'd like to kind of wrap things up, and before doing so, I'd like to kind of pay homage to this German scientist, Hans-Jürgen Stammer, and we kind of wanted to dedicate this new lineage of bacteria to him, as well as to the unique transmission cycle of these beetles. So we're proposing Kandidaten Stammer encapsulation. With that, I'd like to thank a lot of people that were involved in this project, especially the Max Planck Institute for Chemical Ecology, Eugen, Eileen, Roy, Benny, Mike, and Martin Kaltenbrook. As well as Mike Cripps and Takima Katsu in New Zealand and Japan, who opened their labs for this really cool collaboration. And I also like to thank Karina Houghton, who very nicely illustrated